Hello my dear friends, welcome to our YouTube channel pharmajobalert.in. Friends, in this video we're going to see pharmaceutical interview questions for freshers candidates. So either undergraduate or postgraduate freshers, these interview questions are very useful at the time of interview preparation. So let's get started in this video, and before that, if you're visiting our channel for the very first time, do subscribe to our channel for getting regular job updates. My dear friends, coming to the first question, the basic question is, what is solubility? Friends, solubility is a mass of solute that dissolves in a specific volume or mass of solvent at a given temperature. So this is solubility. So for example, 1 gram of sodium chloride that is dissolves in 2.786 ml of water at 25 degrees Celsius. So what he is saying, 1 gram of sodium chloride, here it is soluble in 2.786 ml of water at 25 degrees Celsius. So this is the solubility of sodium chloride. So solubility means the mass of solute that dissolves in a specific volume of solvent at a given temperature. So coming to the next question, define dissolution. Friends, dissolution is a process by which a solid drug substance goes into solution. So solid is will move into the solution. So this is called dissolution. So there are several theories regarding dissolution. So if you are interested, you can learn those things. Those are also very important. The dissolution theory, which is film theory, and so many other Danquart models, so many theories are there regarding dissolution. So please also have a look at those things. So let us move on to the next question, which is, what is pH? So this is another important basic pharmaceutical interview question. pH means negative logarithm of hydrogen ion concentration, which is expressed in mole per liter. So here the negative logarithmic scale is used because the numbers are all less than 1 and vary over a wide range. So lower pH numbers, for example, pH value of 6.2 indicate high hydrogen ion concentration, that is an acidic solution, whereas high pH numbers, that is 7.8, represent lower hydrogen ion concentration alkaline solution. So pH of 1 to 6.2, those indicate the acidic pH range. So in this way, a unit change in pH represents a tenfold change in the hydrogen ion concentration. So one more thing you need to know, a unit change in pH represents tenfold times hydrogen ion concentration. So what is pH? Negative logarithm of hydrogen ion concentration, which is expressed in moles per liter. So let us move on to the next question. So what is pKa? So Ka means dissociation constant. pKa means acid dissociation constant. So here you can see pKa means acid dissociation constant, and it is the negative logarithm of Ka, that is dissociation constant. Stronger the acid, lower the pKa value. It gives degree of ionization at various values of pH. So pKa means acid dissociation constant. So another term is there, which is pKb. pKb means basic dissociation constant, base dissociation constant. So let us move on to the next question. So pKa is very important, and you must also have to learn regarding the Henderson-Hasselbalch equation, and pKa influences the solubility. So it influences the degree of ionization of drug at various pH, and so pKa influences the absorption of the drug. So you need to learn all those things. So pKa means acid dissociation constant, which is a negative logarithm of dissociation constant. So the stronger the acid, it have lower pKa value. So the stronger acid means it is freely soluble, and it gives a degree of ionization at various values of pH. So Henderson-Hasselbalch equation gives the pKa values of drugs, so you can have a look at those by googling them or in any reference textbooks. So next question is, what is Orange Book? Friends, Orange Book is a publication which contains approved drug products with therapeutic equivalence evaluations, which is known as Orange Book. So approved drug products by the FDA with their therapeutic equivalence evaluation is known as Orange Book. So therapeutic equivalence means, for example, there is a brand product. So another generic product which is therapeutically equivalent to it is also mentioned in that book, so which helps in substitution of a branded drug with a generic drug. So Orange Book identifies the drug products which are approved on the basis of safety and effectiveness by the FDA. Friends, in Orange Book, codes are used, A codes and B codes. A codes are used for approved drug products with therapeutic equivalence evaluations, whereas B codes for drug products, FDA still have to determine the therapeutic equivalence evaluations. So that we'll later discuss in the next coming videos. So here for the understanding of yourself, I am saying those things. So friends, this orange book will act as a guide to the public and practitioners like physicians, pharmacists for dispensing of the drug products, therapeutic equivalent drugs. While some states in the US follow the positive formulary and negative formularies. So positive formulary means it contains the drug products which are therapeutically equivalent. So therapeutically equivalent drug products are listed in the positive formulary. In negative formulary, it is the opposite. So let us move on to the next question, BCS classification. So what is BCS classification? Friends, this is a classification based on a theoretical basis to correlate the in vitro drug resolution. The in vitro drug dissolution is correlated with the in vivo bioavailability and was developed by Amidon and the group of scientists. So the approach was based on aqueous solubility and the permeability of the drug through the gastrointestinal tract. So BCS classification was based on the aqueous solubility and permeation of drug through the GIT. And another important thing, the BCS classification was based on fixed first law. J is equal to P into C. The equation J means flux, C is the concentration, and P is the permeability. So the BCS takes into account the three major factors, that is the rate and extent of drug absorption from an immediate release solid oral dosage form, include dissolution, the solubility, and intestinal permeability. So here is a list of classifications of BCS classification. Here you can see 4 classes are there. Class 1 means high solubility, high permeability. These drugs will show high solubility and high permeability. And class 2 drugs show low solubility and they have high permeability. So based on the solubility and permeability, the classification is made. And third one is high solubility, low permeability, and class 4 drugs show low solubility and low permeability. So the main area of interest is class 2 drugs which are having low solubility and high permeability. Most of the drugs will fall under this class 2. So you need to focus on this area. The BCS classification is very important. The next question is, so what are preformulation studies? And friends, preformulation means before formulating the drugs, these studies were performed, and these studies involves the studying the physicochemical properties of the new drug candidates, the physicochemical properties and the new drug that affect the drug performance and the dosage form design. So the properties of the drug that are to be studied during preformulation, mainly for the designing the dosage form and what factors are going to affect the drug performance, to know the factors. So properties of the drug that affect the drug performance and as well as the dosage form design. So coming to the next question. So in the next slide, we are going to learn about the major area of research in preformulation. Before that, let us have a look at the types of dissolution apparatus. So what are the types of dissolution apparatus? Friends, there are 7 types of dissolution apparatus are there. The apparatus 1 is rotating basket type, and the apparatus 2 is paddle method, and the apparatus 3 is reciprocating cylinder, apparatus 4 is flow through cell, apparatus 5 paddle over disc, apparatus 6 cylinder, and apparatus 7 reciprocating disc. So please remember all these apparatus in a sequence. So these are very important from the interview point of view. So the apparatus 1 is rotating basket, apparatus 2 rotating paddle, apparatus 3 is reciprocating cylinder, apparatus 4 is flow through cell, and these 4 are very important. Then apparatus 5 is paddle over disc, apparatus 6 cylinder type, apparatus 7 reciprocating disc. So coming to the next question, the major area of preformulation research. So what are the major area of preformulation research? So these are the major areas which include bulk characterization, solubility analysis, and stability analysis. Bulk characterization, which include the crystallinity and polymorphism, hygroscopicity, particle characterization, powder flow properties. And next, solubility analysis include pKa, ionization constant, pH solubility profile, the common ion effect, solubilization, dissolution, and partition coefficient. Coming to the stability analysis, which include solution stability, pH rate profile, solid state stability, bulk stability, and stability in toxicology formulation. So these are the major area of preformulation, which include three major areas: bulk characterization, solubility, and stability analysis. So coming to the final question of this series of pharmaceutical interview questions for freshers. So what do you mean by quality by design? So you might have heard this word many times in your B.pharma also. This is in a way emerging. So the pharmaceutical quality by design is an approach for the development that begins with predefined objectives. The objectives are predefined and emphasizes product and process understanding and process control, which are based on the science and quality risk management. So quality by design is an emerging to enhance the assurance of safe, effective drug supply to the consumer and also offer promise significantly improve the manufacturing quality performance. So by quality by design, we not only get the quality products, we can also reduce the unwanted costs. So improving the manufacturing quality performance. So these are the complete interview questions in this video. So I hope you like these questions and the information posted by us in this channel. So for getting continued updates like this in our channel, subscribe to our YouTube channel and also click that notification bell icon. You can also share this video with your friends so that they can also get benefited.